There are living light shows made of hundreds, sometimes thousands of tiny bodies, all blinking together like an alien starship cruising through black water. And the wildest part? None of this is science fiction. These are your neighbors, just several thousand meters downstairs. Welcome back to Exploding Critters. Today, we're introducing our new series called Under the Sea. In this series, we'll be heading straight into the deep, the Twilight Zone, the Midnight Zone, and beyond. A world with no sunlight, crushing pressure, glowing hunters, slow-motion monsters, and some of the strangest real animals evolution has ever signed off on. After this, you will never look at the ocean or the idea of aliens the same way again. The planet's deep ocean is a realm that remains as mysterious as the surface of the moon. At depths below 200 meters, sunlight vanishes, and the world becomes an endless expanse of black water with crushing pressure, near freezing temperatures, and almost no food. Yet this otherworldly environment is not barren. It is filled with organisms whose adaptations read like science fiction. Pressure here can exceed 1 ton per square centimeter, a pile of elephants pressing on each square centimeter of your body. But life has not merely survived. It has thrived and diversified into forms and behaviors that challenge our assumptions about biology. To explore this world and its inhabitants, we must understand the physical challenges, the evolutionary innovations that conquered them, and the modern technologies that allow humans to peek into the abyss. At around 1,000 meters, water pressure is about 100 times greater than at the surface. By 6,000 meters, it is 600 times greater, and in trenches more than 8,000 meters deep, it approaches 1,000 times atmospheric pressure. Such pressures would crush gas-filled cavities and deform conventional skeletal structures. Instead of armor, deep-sea animals often use softness and chemistry to survive. Many deep creatures have gelatinous bodies, flexible or reduced skeletons, and avoid gas-filled organs. Soft tissues and high water content help equalize internal and external pressures, preventing collapse. Fish that in shallower water use buoyant swim bladders to stay afloat have replaced them with fatty livers or low-density bones. Cellular membranes are tuned with special lipids that remain fluid under pressure. In some deep-sea comb jellies, 3/4 of the membrane lipids are plasmalogens. These cone-shaped molecules are bent, allowing membranes to remain sturdy and flexible under crushing pressure and preventing them from splitting apart. The adaptation, termed homeocurvature, helps membranes maintain their shape and function at extreme depths and is separate from adaptations to cold. Among the vertebrates, the champions of pressure adaptation are the hadal snailfishes. These fish live thousands of meters down in trenches like the Mariana. Anatomically, they look like pale tadpoles, yet they hold records for depth. Researchers examining their anatomy and genetics found that snailfish skulls have gaps between bones, and much of their skeletal material is cartilage rather than calcified bones. A mutation in a gene responsible for bone hardening reduces calcification, making the skeleton flexible. A complete rigid skull would crack. Thus, the gaps allow internal and external pressures to equalize. Snailfish tissues carry high concentrations of trimethylamine oxide, TMAO, a molecule that stabilizes proteins under pressure. Whereas most animals have 1 copy of the TMAO synthesizing gene, these fish have 5. They also produce large numbers of transport proteins to move substances across membranes and have lost functional eyes because photoreceptor genes are unnecessary in total darkness. Genetic analyses show they retain a Rh1 gene for monochromatic vision, but have lost genes for seeing other wavelengths, while auditory genes are preserved and circadian clock genes are largely absent. A pseudogene for GPR27, involved in metabolism, may reduce metabolic rate and missing bone mineralization genes leave skulls partly open. Recent genomic studies suggest that pressure tolerance in deep-sea fish may also involve convergent mutations in the RTF1 gene. This mutation increases transcription efficiency and may act as a newly discovered genetic mechanism for pressure adaptation. Other species, such as comb jellies and E. coli engineered with plasmalogens, show how lipid chemistry can confer pressure resistance. The general pattern in these creatures is to avoid gas spaces, rely on cartilage or gelatinous tissues, use piezolites like TMAO, and adjust membrane composition to preserve fluidity under pressure. Below 200 meters lies the mesopelagic zone, where light from the surface dwindles to blue gloom. By 1,000 meters, it is virtually gone, so no photosynthesis occurs. Without sunlight, plants and algae cannot grow, and primary production at these depths depends on two main sources: marine snow and chemosynthesis. Marine snow is a continual rain of organic debris, dead plankton, fecal pellets, mucus, pollen, and dust that sinks from productive surface waters. These flakes slowly descend over weeks, growing as they collect particles and providing food for filter feeders, scavengers, and predators in the deep. Occasionally, larger carcasses such as fishes, squids, and even whales sink, creating temporary buffets in this zone. Whale falls undergo a series of stages. Large scavengers strip soft tissues within months. Enrichment opportunists and microbes consume the remaining detritus for years. Finally, bacteria break down lipids inside bones, releasing sulfide and feeding chemosynthetic organisms. These falls support communities for decades and link surface productivity to the abyss. Chemosynthetic ecosystems form where geology provides energy rather than sunlight. At hydrothermal vents, seawater percolates into the crust, heats to hundreds of degrees, reacts with basaltic rocks, and returns loaded with chemicals like hydrogen sulfide. Microbes living at vents oxidize these compounds to make carbohydrates, forming the base of entire food webs. Giant tube worms, mussels, clams, and shrimps host symbiotic bacteria in their tissues. The bacteria provide nutrition in exchange for a safe habitat. Vent shrimp possess special heat-sensing organs and feed on bacterial mats. Vent mussels house chemosynthetic bacteria in their gills to convert inorganic chemicals into sugar. Cold seeps, where hydrogen sulfide and methane seep through seafloor sediments at ambient temperatures, host similar communities of clams, mussels, and tube worms. In brine pools, seawater dissolves underlying salt layers, creating supersaline lakes on the seafloor. These pools are often anoxic and toxic to most life, yet along their margins, microbial mats and mussels flourish by using chemical energy. Shrimp hover around the edges, using the dense brine to trap prey, and amphipods and small sharks patrol the boundaries. The brine preserves organic material, creating archives of past climate. Even in open water, energy is scarce. Many deep-sea animals have very slow metabolisms and long lifespans. Greenland sharks grow less than 1 centimeter per year and reach sexual maturity after more than a century. Radiocarbon dating of eye lens protein suggests individual sharks may live 272 to 512 years. Pacific sleeper sharks and other deep sharks also have slow growth, late maturity, and low reproductive rates. Such slow life histories make them vulnerable to overfishing, yet they are part of the deep-sea mosaic of longevity and patience. With little food available, many deep-sea fishes have large, expandable stomachs and jaws, enabling them to eat meals of almost any size when opportunity occurs. They drift rather than sprint, saving energy, and adopt sit-and-wait strategies instead of long chases. In the absence of sunlight, many deep-sea animals produce their own light. Bioluminescence is a chemiluminescent reaction where a substrate, often called luciferin, is oxidized by an enzyme luciferase, releasing energy as photons with little heat. Different organisms have evolved distinct luciferines and enzymes. The presence of multiple systems suggests bioluminescence evolved independently many times. Fish, squids, jellyfish, crustaceans, and bacteria produce blue or green light. Some dragonfish emit red light invisible to most prey. The functions of bioluminescence are diverse. Counterillumination matches downwelling light to erase an animal's silhouette. Burglar alarms use a startling flash to deter predators or attract larger predators to attack the attacker. Lures attract prey, such as the glowing lure of anglerfish. Deep-sea animals have also evolved extraordinary eyes and sensory systems to make use of scarce photons or other cues. The barrel-eye fish, Macropina microstoma, is a small, hovering fish with a transparent dome over its head. Beneath this dome are two bright green tubular eyes that normally point upward to scan for silhouettes of prey drifting above. When the fish needs to feed, these eyes can rotate forward to track the prey as it moves into the fish's mouth. Its large, flat fins allow precise maneuvering, conserving energy, and although it has a small mouth, it possesses a large digestive system to process small drifting animals and pieces of jellyfish. Lanternfish are among the most abundant vertebrates on Earth. Over 245 species in the family, Myctophidae collectively make up around 60% of all deep-sea fish biomass. Their bellies and sides are covered with light organs called photophores. The arrangement and flashing pattern is unique to each species and may play a role in camouflage and communication. Most lanternfish migrate to the surface at night to feed on zooplankton and return to the depths by dawn. Their movements were once misinterpreted by sonar as a false bottom because of the enormous numbers. Lanternfish regulate buoyancy with gas-filled swim bladders, a rare adaptation among deep-sea fishes. The deep-sea anglerfish is famous for its glowing lure. In the meso and bathypelagic zones, female anglerfish possess a modified dorsal fin spine that supports a bioluminescent organ housing bacteria. They dangle this lure to attract prey within range of their huge jaws and needle-like teeth. But the anglerfish's most extraordinary adaptation is its reproductive system. Males are tiny compared to females and act essentially as mobile gonads. When a male finds a female, he bites and fuses to her body, connecting their circulatory systems. Over time, he degenerates into little more than a pair of testes. Some species carry multiple males. This sexual parasitism ensures that when a female is ready to spawn, sperm is available in a world where encountering a mate might otherwise be impossibly rare. The adaptation evolved along with features like the bioluminescent lure and large jaws, enabling anglerfish to colonize the deep sea. The gulper eel, Sacchopharynx lavenbergi, is a midwater predator with a head and jaw that opens like a balloon, far larger than its slender body. Its expansive mouth and elastic stomach allow it to swallow prey of almost any size that drifts into its path. The tip of its long whip-like tail glows, possibly acting as a lure. In an environment where meals are few and far between, volume-oriented feeding strategies are advantageous. The bigfin squid, Magnapinna spp, remains among the most enigmatic deep-sea creatures. Fewer than a dozen sightings exist, but observations reveal huge fins and extremely long arms and tentacles that hang down like marionette strings. The arms can reach over 6 meters, about 20 times the body length. The squid can live deeper than almost any other cephalopod, with one recorded at 735 meters. Scientists suspect the sticky appendages capture small prey that bump into them. Only 3 species have been formally described. More probably await discovery. These characters exemplify the inventiveness of evolution when faced with pressure, darkness, and starvation. Their forms may appear alien, but each feature is a solution to a specific environmental constraint. Recent genomic studies have begun to unravel the evolutionary pathways that led vertebrates into the deep. A 2025 multi-omic study spanning depths from 1,218 to 7,930 meters collected 11 deep-sea fish species and reconstructed their evolutionary history. Researchers identified 2 distinct pathways: ancient survivors, lineages that colonized the deep sea before the Cretaceous mass extinction, and new immigrants, species that entered the deep after the extinction. The study confirmed that TMAO levels increase with depth up to about 6,000 meters, but beyond that, the trend does not continue. Instead, a highly conserved mutation in the RTF1 gene appears across all fish living deeper than 3,000 meters, enhancing transcription efficiency and representing a newly discovered pressure adaptation mechanism. Such insights challenge the traditional TMAO hypothesis and illustrate convergent evolution at the molecular level. The same study revealed high concentrations of PCBs in hadal snailfish livers, demonstrating that human pollutants have reached the deepest trenches. This combination of evolutionary discovery and environmental warning underscores the need for interdisciplinary deep-sea research. In the next videos, we will be diving into a deep research about each underwater creature, so make sure to stay tuned and hit that like and subscribe button to be updated when the video comes.